Hey kids, what's up? Mr. Brotherton here. We're going to be talking about proteins. Now we are really hitting the big time. This is the super exciting stuff. I love proteins because they are the doers of life, and everything you're pretty much looking at here is a protein. So what you can see of our bodies is usually proteins. After water, the largest component of life is going to be protein and then DNA. And what's cool about proteins is their structure is directly related to that genetic information, because that genetic information dictates the structure of these proteins. So what's also really cool about proteins is we can look at evolutionary relationships using proteins. One example of that is this guy right here. This is cytochrome C. Cytochrome C is used in the electron transport chain. So when we're doing our metabolic processes to generate ATP, this little guy right here is part of that process, kind of shooting electrons through the ETC to help generate ATP. What's really cool about cytochrome C is it is a very highly conserved protein across vastly different species on planet Earth. Okay, so it's only 104 amino acids long, and 37 of those are never changed. So of all the samples we see across totally different animals, 37 of those never change. Our cytochrome C looks exactly the same as a chimpanzee cytochrome C. We're only one amino acid off from a rhesus monkey. Our cytochrome C is very similar to even something like a yeast, which is a fungus, which we diverged from a long time ago. So we can actually look at evolutionary relationships through proteins. Another really cool thing about proteins is they kind of can also show us that there may be a limited number of ways of solving similar problems. So if you actually look at the cytochrome C here, this little dude right here, this is known as a prosthetic group, and we'll kind of get into that towards the end of this, but it's an extra little peptide here, extra little protein here, that is used to kind of usher electrons through here. And if you look here, this thing is known as a heme. Guess what? This up here is a hemoglobin, and it's made of 4 of these. What's really cool about this ring structure here is you will see that exact same structure again when we talk about photosynthesis, because it looks almost exactly the same as chlorophyll, except chlorophyll has a magnesium here instead of an iron. What's also really, really cool about this heme or chlorophyll is it actually looks very similar to the same type of molecule, the same type of protein that's going to be used by life that has a sulfur-based metabolism. So if we think about the very earliest forms of life on Earth, they were probably going to be anaerobes near hydrothermal vents and doing this kind of sulfur-based metabolism. And guess what? They have a very, very similar molecule. So both chlorophyll, heme, and the molecule that these sulfur dudes are using, it all looks very similar. So that's also showing us there may only be a limited number of ways of solving the same problems, but it's also showing us variations on a theme and these relationships that proteins represent, which is really incredible. All right, so proteins, oops, I'm too excited. Proteins are the most structurally and functionally diverse group of biomolecules. All right, these things are super, super complicated, and they're involved in everything. All right, enzymes, structure. Our hair is going to be made of keratin. Ligaments and stuff are going to be made of collagen. They're going to carry things. They're going to be used as receptors and binding. So defense, we can actually use proteins to attack viruses. They're going to be used for muscle contraction. So like right now when I'm moving my arm, I'm using actin and myosin. We can use proteins as hormones, so for cell signaling. We can also use proteins to store things like nitrogen that need to get later used for other metabolic processes. And we'll actually see some of these amino acids end up getting used for other things later on in this course. All right, so proteins, just like everything else, a protein, what we're really talking about here is this polymer, okay, of these simpler things. So the monomer, mono means one, our monomer here is going to be an amino acid. There are 20 different amino acids that most life uses. There are more than that, but there's really only 20 we're concerned with. And the polymer of that is going to be called a polypeptide. The reason for that is because the bonds that we're making here are known as peptide bonds. So when we stick two amino acids together, we're making a peptide bond. So a polypeptide is a bunch of amino acids stuck together. So let's think about this number 20. Okay, so if we want to make this guy down here, which would be known as a dipeptide, okay, because we've stuck two amino acids together, if we look at this, there are a lot of different combinations that can be happening here just by making a dipeptide. So if you imagine you have 20 amino acids potentially, then you have 20 more that you could stick together just to make a single dipeptide. So the possibilities just by sticking two amino acids together are going to be 20 times 20. That's 400 different possibilities. If we stick 3 amino acids together, that's going to be 20 times 20 times 20. All right, now we're looking at 8,000 possibilities here just by sticking 3 amino acids together. All right, what's crazy is something like insulin is relatively not that complex. It's only 51 amino acids. That cytochrome C we were talking about is like 105, 104 amino acids. Something like titin, which is a muscle protein, is 34,000 amino acids. So if we think about something as simple as insulin, maybe around 100 amino acids, that would be 20 times 20 times 20 times 20 times 20. Okay, so it's 20 raised to a power of 100. That is an absolutely astronomically large number just by combining 100 amino acids together. The potential there, it's basically infinite. How big is the number 20 raised to the power of 100? It's more than the electrons that exist in the entire universe. Okay, that's crazy. So proteins are incredibly diverse simply because there's so much possibility here of sticking things together. So what is an amino acid? It's basically a central carbon group here, this amino group here, and a carboxyl group here. So this guy right here is going to be a base. This guy right here is going to be an acid. So they basically neutralize each other. So we have a pretty neutral pH. And then finally, you have this R group, that side chain, and this is what's going to give us that really big diversity that I was just talking about. Okay, this is what's going to give it those unique chemical properties. So what's really cool about amino acids is some of them, they all have different properties, right? So these are going to be our nonpolar and hydrophobic amino acids. Why are these nonpolar and hydrophobic? Well, as you can see here, they're mainly made of hydrocarbons, and they're not going to be polar, all right, because their electronegativities are relatively balanced and they don't have things hanging out that really want to bond with stuff. So if they're nonpolar and hydrophobic, you're mainly going to find these amino acids on the interior of proteins, or you might find them on the interior of membranes, so inside of our phospholipid bilayer. So if we check out these polar amino acids, these guys are polar. They do not have a charge though, so they're polar and uncharged. Well, how is that possible? It has to do with the fact that it kind of has this dipole where overall this whole thing is not charged, so our positives and negatives are balanced, but there's still a little bit of stealing of electrons going on, so you get these kind of partial charges, all right, kind of in the way that water is kind of partially charged. Whereas if we go down here, you have these electrically charged polar amino acids, and these guys are pretty much going to be hanging out towards water, oriented towards water. I'm going to dive more into this acidic versus basic thing here because this made no sense to me for the longest time. I'm like, how is this acidic? Acidic things donate protons, so they should have an H hanging out here, and I don't understand how these things are basic. Because basic things like to suck up a hydrogen. So we're going to kind of talk about what's going on there. So why are these polar and hydrophilic? Because these ones up here are partially charged and these ones down here are definitely charged. Okay, so what's going on with these amino acids that are acidic? Well, the thing is at the pHs that we're talking about in life, when we look at these amino acids, we're actually showing them after they've already donated their hydrogen. So when we look at aspartate, I was like, how is this thing an acid? Okay, because it's got this negative charge. That makes no sense to me. But we're actually showing it after it's kind of already done that chemical, okay, that chemical reaction. So we can get rid of that H and we can get rid of that H. All right, and now this thing is charged. So if you can kind of think, this hydroxyl has already done its reacting. That proton has already been donated. That's how we tend to show these things. And these over here are not acidic. The reason they're on this slide is, if you notice here, all we've done is we've substituted this NH2 where this oxygen was, and they basically have the same exact structure, except this little group is different than our oxygen. So when we use the term derivative here, it means that we've just replaced this guy with this guy over here. So these guys are not acidic. In fact, you would probably think that they would be basic based on this group here, but there's weird little interactions happening here between this nitrogen and this carboxyl kind of group. So there's stuff happening there that makes this not basic, okay? But that's a little bit more than you need to know. Just know that only 2 amino acids are in fact acidic. All right, so what's going on here with these amino acids that are basic? They have these basic side chains. Well, we're showing them actually after they have already accepted a hydrogen. So one definition of a base is something that can accept a proton. And what we're showing here is actually after these have already accepted their proton and now they have a positive charge. So this guy's already taken in a proton. This guy has taken in a proton. And then histidine right here has also taken up a proton. What's really, really cool about this acid-base thing that's going on here and giving these things charges is, guess what? The shape of our protein can change, because now we have positively charged parts of our protein because of these amino acids, and we have negative parts. Another really cool thing, especially about histidine, is that this thing is going to be found a lot of times in the active sites of proteins and enzymes, because it's pretty good about giving up its proton here. So right here it's in its protonated form, so it's taken on a proton, but a lot of times you'll see this thing deprotonated where it's given up its proton, so that way it can start doing some really cool binding, and we'll get into that later. Another cool thing, cysteine can form these things called disulfide bridges, which is basically this sulfur can react with another sulfur group, and the hydrogens will go off and form their own bonds, and then the sulfurs will be covalently bonded to one another. So if you've ever wondered why perms smell like rotten eggs, it's because you're adding this ammonia solution that breaks. It actually is breaking these sulfur bonds, so that's why you smell that sulfuric smell, and then you're reforming them in the way you want them to be. So keratin is mainly made of a lot of these cysteine disulfide bonds, and when you break them, it stinks. Okay, so what's going on in a peptide bond? Once again, dehydration synthesis, it's coming back. So we take this hydroxyl here, we take this hydrogen here, we stick them together, water goes away, and now you have a peptide bond. So this is how we can make these really long polypeptides just by sticking these things together. All right, so peptide chains, we talk about an N-terminal and a C-terminal, and it's basically kind of the way we read these things. So if you look here, this is going to be the N-terminal because it's the amino group, so it has the nitrogen, and then the end of it is going to be where that carboxyl is. That's going to be the C-terminal because that's where that carbon group is going to be, okay? These things grow in one direction due to protein synthesis. So if we go all the way back, the central dogma of biology is DNA makes RNA makes protein. Well, in that making protein, we have a ribosome that's sticking all these amino acids together, and so they're going to grow in one direction, okay? And so there you go, you can see that. So protein structure and function, once again, this is the kind of where that idea of structure dictates function really, really comes home to roost. This is incredibly, incredibly important, all right? So that 3D structure, we have all these different interactions that can happen. So let's kind of dive into that. So the function is going to depend on the structure, and it all starts with that order of amino acids. And what determines the order of the amino acids? Well, that's DNA. Okay, so the reason we can look at proteins and kind of determine evolutionary relationships is because that order is determined by DNA. What's cool is DNA breaks down pretty quick, but proteins are relatively stable, so we can actually extract proteins from fossils and we can compare them to one another, and we can start to figure out evolutionary relationships for things that are really long dead without having to do some phylogenetic research, okay, which is really, really incredible. All right, so protein structure is determined by 4 different levels. Okay, so the very first thing is known as the primary structure. This is determined by the sequence of DNA. This is what a polypeptide actually is. So when we talk about a protein is kind of after this thing is done folding into all these different structures. But this first primary structure, this is determined by the amino acid sequence. And this is actually what when we say polypeptide, this is what we mean, just that long initial chain, okay? And if you change one of these, it can make a huge difference. All right, so if you were to change one of these, you're going to change the further chemical bonding. A really good example of this is the DMD gene, which makes dystrophin. If you mess up that DMD gene just in one single place, it actually adds a stop codon in there, and you actually cut your protein in half, and that can cause muscular dystrophy. So here's another example, sickle cell anemia, which is made of those 4 heme proteins stuck together that we talked about earlier. This one single substitution here is going to make this thing from this shape, okay, into this shape. So the role of hemoglobin is to carry oxygen and carbon dioxide, and this thing is not as good at it, which can cause problems. So the secondary structure is the local folding. So this is folding along short sections of our polypeptide. It's basically happening due to hydrogen bonds between the oxygen and the H of different amino acids. So there's 2 different ways this happens. You can have what's known as these alpha helixes, or you can have these things known as beta pleated sheets. So an alpha helix is where our oxygen and hydrogen are bonding to each other on every 4th amino acid. And so this kind of looks like a really old school phone line, like one of those old telephone lines that's all curly. Or you can have something happening like this where 2 different polypeptides kind of meet up together, or it folds back on itself and you get this beta pleated sheet. And so that structure is going to look like maybe a Venetian blind or a pleated skirt. So that's our secondary structure, and you can kind of see more of that here. And our tertiary structure, this is going to be where the whole molecule starts folding. So this is where you get really complicated interactions between the R groups. So those groups that we were talking about coming off the side. So those acids, those bases, that cysteine, which is going to create these disulfide bridges. So you can start to form these ionic bonds between those acids and bases, which is, going to be pretty strong. And then you have these covalent bonds between our disulfide bridges. And then you can get these hydrophobic interactions, which, while not true chemical bonds, the hydrophobic parts are going to move towards the inside of our folded protein. Okay, so all of that makes a difference because the structure is going to determine how this thing interacts. All right, so our quaternary structure, not all proteins are going to have this quaternary structure. This is where you take more than one polypeptide chain and you stick them together, okay? And then only then is it a functional protein. So hemoglobin is actually made of 4 different heme polypeptides. So you stick all those guys together and that's how you get a hemoglobin. Or in the example of collagen or these like really fibrous type of proteins, you got to stick a bunch of them together to make these quaternary structures. So this is 2 polypeptides stuck together. So here's our review. First happens is the peptide bonds. The second thing that happens is those R groups can kind of interact to form those hydrogen bonds. The tertiary structure is where you get really crazy interactions between our R groups, and the quaternary structure is many polypeptides stuck together. You can also have these things called chaperonins or chaperone proteins that help with protein folding. We used to think that it was entirely determined by the DNA that made the protein folding happen, but actually for them to fold correctly, sometimes they'll go inside these things called chaperone proteins that create certain conditions where only certain chemical reactions will happen to make sure that the protein ends up correctly folded. All right. we use a lot of different protein models, but we don't really need to go into that right now. Mainly, we use computer modeling right now. All right, guys. So that's all.